syllabus by term 3. So in term 4, when we come back, you really do revision and your year-end exams. So essentially, this is our last term together for biology. These are the topics that will be covered for term 3. I'm going to start with a quick revision of cell division. Please ensure that your SLS is completed, then goes into your EPSR. Okay, I'd like to collect also your reflection worksheet. It is a green worksheet. Yeah? By next week, can? So next week, the first lesson I'll have with you is on Thursday. So Thursday is when I'll collect, and then we'll also go into this. Yeah? It's not a small amount of days, 15%, so it's quite significant. Okay? After cell division, we will then go to the next topic, reproduction. Okay, that will begin next week. So all of the necessary materials are already available. You can read up first beforehand, you can look at the SLS. If you want to get a head start. After reproduction, then we will go into the next chapter called inheritance. This is where we will learn about how you inherit all these characteristics that you have within you on the surface from your parents. Last but not least, we end up this year with genetic engineering. Once we know how reproduction goes and how we pass on characteristics from one person to another, and we have the ability to control this process, genetic engineering. We aim to complete all of this by term 3. Currently, we are going to do a quick revision on cell division. More specifically, today I want to spend a quick amount of time to revise a few things. Number 1, mitosis. Okay? And how to identify stages. From microscope. Number 2, we will revise meiosis. And then that's about the revision I will do from what you have studied in your SLS. After that, we will dive into reproduction topic. Okay? First and foremost, may I get you to open up your digital notes for cell division. Go to the section of mitosis. We'll do a quick revision. On the whiteboard, I'm going to put up some magnets later on. We will use this to do a quick revision also. Maybe we start off with a quick overview on the cell cycle. This was where we left off before you went out for your holidays. Can you still remember all the different phases? Assuming it goes in this direction. Okay, then we do a quick revision. I call you bit by one by one, then you give me whatever you can. Okay, let's slowly begin. Maybe we start the story first with this. How many chromosomes do humans have? Okay, how many chromosomes do humans have? I have 6 for 46. We have 46 chromosomes. What does that actually look like? Okay, let's look at a single chromosome first. What in the world is a chromosome? Okay, a chromosome is made up of DNA. DNA is double-stranded. Yeah? That means to say, if I were to look at a chromosome looks something like that. Here I have a chromosome, and it is double-stranded. You following? So we have 46 of these. Imagine you have this, but you go and times 46. Another way to look at this is that other than us having 46 chromosomes, actually we have 23 pairs of chromosomes. What do we mean by having 23 pairs of chromosomes? This is what I mean. Oh, I think a quick Google image will be able to give you the answer. 46 chromosomes. Okay, humans have 46. We have more specifically 23 pairs. I'd like you to look at these pairs. Notice that what makes a pair is that lengthwise, size-wise, they are the same. Okay, granted, some of them are a bit bent, but if I straighten it out, actually they're the same. I will not pair the short one over here with the long one up there. Okay, humans, we have 23 pairs of chromosomes. And each pair, if you look at them, same size, same length. And actually they contain the same genes. What do I mean by that? If I were to look at this pair over here, this is perhaps where you'll find the genes, the DNA that is in charge of making your eye color. Maybe you go to another pair. This pair over here may contain the genes that define your hair color. Maybe you go to another pair. You could perhaps find the genes related to your skin color, so on and so forth. Okay? Each pair consists of the same genes that instruct a certain part or characteristic of your body. So that's the first piece of information. Let's go and look at the cell cycle now. Imagine we look at a human cell cycle. We have 46 chromosomes, yeah? 46 chromosomes. 23 pairs. Okay, let's begin first with this large phase over here. What's this phase? Okay, G1, or in other words, Gap1 phase. After Gap1 phase, we go into S phase. S stands for synthesis. To synthesize what? Okay. What is this synthesis for? In this particular phase, we synthesize DNA replication. Okay? DNA replication. What does this mean for our DNA? If we have 46 chromosomes, by the end of DNA replication, how many chromosomes will we have? Are you doing the math? This is a trick question because there's still 46 chromosomes. How is that possible? I'll show you why. Imagine now I extract out just one of these chromosomes, okay? You follow along with me. Let's just take out one of these chromosomes. Okay, one of these chromosomes. Okay? After S phase, we will replicate the DNA. So I should have two copies now. So other than one copy, I'll have two copies. Following? Okay, the interesting thing is, our body, our cells, won't let the copy just fly away. Instead, we will ensure that the copy stays with the original copy over here and attach itself to a point. We call this point over here a centromere. Okay? So after S phase, you'll find that the copies of the DNA will be attached together at the centromere. Okay, so what about these, right? Okay. Earlier on, I said there are 46 chromosomes, right? 23 pairs. Okay? This is just one of the pairs. Let me draw in the second part of the pair. So imagine two pairs, right? So here I have two pairs. Number one, number two, just like what you see here. After S phase, this is what you should see. Following? So if I were to extrapolate this, let's apply this to all of these chromosomes. This is what you will see. Okay. After S phase, you should see then every single pair will now have a copy. And they'll be attached together usually at a single point. And we refer to that as a centromere. I show this to you because if you were ever to see a structure that remotely looks kind of X-shaped, it means that DNA replication has already occurred. If instead someone were to show you a diagram that looks like that, okay, this one over here, this implies that DNA replication has not occurred yet. Okay, this one, it has already occurred. So if you like to, you can even Google, find these two images, and paste them into your textbook. What did I type into Google? I typed 46 chromosomes. And then I get two different versions of images. One version just shows me pre-S phase, and another version shows me post-S phase. Yes. Oh, okay. Okay. Sorry, I didn't answer the question. So just now I said before S phase is 46, after S phase, still some people will still describe as 46. Okay. Why? Because we still consider one of these entities as a single chromosome. Okay. As long as they are attached together, we consider it as one unit. Okay. So even though we have two attached to each other, is still a single chromosome. Okay, a single unit. So if you want to test your understanding a little bit more, okay, let's have a look. Here at the very beginning, I have 46 chromosomes. Can you tell me how many DNA, listen carefully, how many DNA strands are there in 46 chromosomes? Okay, how many DNA strands are there in 46 chromosomes? Assuming each chromosome is made up of 2 strands. Double helix. So you know the math, equals to, so there are 92 strands. Following? However, after we go through S phase, even though I still have 46 of these entities, how many DNA strands do I have? I have actually twice the amount of DNA strands. Yeah? Following? Okay, yeah? Oh, when do you call, what is a sister chromatid? Okay, I'll come to you in a while. Okay, so following. So what has changed? We have the same number of chromosomes, but what has changed is that actually I have 92 times 2 now DNA strands in total. Following? Finya asked, what is this word? You may have encountered it while learning something called sister chromatid. Okay? What is a sister chromatid? A sister chromatid refers to just one of these halves of the chromosome. Following? So another way to phrase it would be, let's look at this chromosome here. How many sister chromatids are there? There are 2 sister chromatids. This is sister number 1. This is sister number 2. Okay? So that's how you can use the words. A single chromosome has 2 sister chromatids. attached to each other at a single point called the centromere. Or just vocabulary. So then we test your understanding. You look at the 46 chromosomes here. How many sister chromatids are there? 46 times 2. Yeah. Every chromosome has 2 sister chromatids attached to each other. Okay. Okay. One last thing I'd like to define for you. Something special happens between here to here, between prophase to metaphase. And that process is condensation. You have learned by now that in the process of mitosis, there's a lot of separation of chromosomes into different cells, correct? But have you ever tried an untangling before a bundle of string? If I ask you to know many different strings, I ask you to pull them apart, chances are they'll get tangled up. The cell has devised a very unique way of separating these strings of DNA without any tangling taking place. And the way they go about it is, after S phase has occurred, they go through one more step during prophase. After S phase has occurred, let me pull this up. This is the class where if I don't stop this, not come back down, right? Okay. After S phase, okay, during prophase, this next thing happens. Rather than let the DNA be so uncoiled and so long, it will compress it and condense it during prophase. So let me condense my DNA until it is now very compressed. So this is one sister chromatid, this is another sister chromatid. I'm trying to represent that I've condensed it. Okay? And here again, I've condensed the DNA, one sister chromatid, another sister chromatid. Okay? This happens during prophase. I'd like to represent this now, from now onwards, using my magnets. So this is a 1, this is a 2. Okay? From now onwards, every time you see my magnets, I'm representing a very condensed version of these structures. Okay, a very condensed version of these structures. Every time you see this from now onwards. We call these structures metaphase chromosomes. It is named after the phase where you can start to see these structures. Okay? After prophase, then you will start to see these very nice butterfly structures. Before metaphase, you won't really see these structures yet. They are starting to form, but not yet. It is when you hit metaphase that you start to see these structures appear. Okay, I'm showing you all this so that later on, as we go through the phases, you're a little bit more aware of why we represent things certain way. Yes? Oh, okay. You don't ask, do the DNA strands, these double-stranded DNA, right? Do they like separate themselves when they condense? They don't. They still remain attached to each other, but they condense and like kind of compact. Just like how you would, a ball of yarn, you won't just toss the whole yarn, right, uncoiled into a box. You will coil it around maybe a tube, and you will put it neatly arranged inside the box. This is what your cell is trying to do before it starts to separate the DNA into different cells. process?
In this lesson, we'll be exploring the M phase of the cell cycle, including mitosis and cytokinesis. Let's do a quick review of the cell cycle to see where they fit in. The G1, S, and G2 phases make up interphase. And the M phase represents cell division. Cell division includes division of the nucleus, called mitosis, and division of the cytoplasm, called cytokinesis. Mitosis is further broken down into 4 phases: prophase, metaphase, anaphase, and telophase. Prophase is the longest phase of mitosis. Prophase is when chromatin begins to condense into the shape of chromosomes, and the nucleolus disappears. The previously replicated DNA coils tightly into sister chromatids. For the first time, you see individual chromosomes. In the center of each chromosome, the centromere attaches the sister chromatids together. Meanwhile, in the cytoplasm, microtubules, known as spindle fibers, begin to fan out from 2 sets of paired structures, called centrioles. The spindle fibers elongate as the centrioles begin moving to opposite sides or poles of the cell. While this is happening, the nuclear membrane surrounding the nucleus disappears. Now that chromosomes are no longer separated from the cytoplasm, the opposite ends of the spindle fibers can attach to the centromeres. Next, the cell enters metaphase. The centrioles complete their movement to the poles of the cell, while the spindle fibers line up the chromosomes along the equator of the cell. The end-to-end alignment of the chromosomes results in a sister chromatid on either side of the equator. Anaphase follows metaphase. During anaphase, spindle fibers separate the sister chromatids at their center. Once separated from each other, each chromatid is called a chromosome. The single-stranded chromosomes form a V shape as the spindle fibers shorten and drag them through the gel-like cytoplasm. The chromosomes move to opposite poles of the cell toward their centrioles. It's common to confuse centrioles with centromeres, which connect chromatids. Remember, centrioles are at the poles. Telophase is the final stage of mitosis. In telophase, the nuclear membrane reforms around each set of chromosomes. Then, the chromosomes spread out into chromatin, and the nucleolus becomes visible once again. Mitosis, the division of the nucleus, is now complete. The final step of the M phase is cytokinesis, the division of the cytoplasm. In animal cells, cytokinesis occurs through the inward movement of the cell membrane. This progressively pinches the cytoplasm until 2 identical daughter cells form. In contrast, plant cells can't pinch in 2 because they have a rigid cell wall surrounding their cell membrane. Instead, cell wall material assembles along the equator, forming a structure called the cell plate. The cell plate grows until it joins with the existing cell membrane, separating the 2 halves of the cell into daughter cells. Over time, new cell walls form between the 2 daughter cells. Here are the key points to remember. The M phase is the 4th and final phase of the cell cycle. During the M phase, cell division occurs through 2 processes: mitosis, when the nucleus divides, and cytokinesis, when the cytoplasm divides. Mitosis has 4 phases. During prophase, chromatin condenses into chromosomes. spindle fibers form, and the nucleolus and nuclear membrane disappear. During metaphase, spindle fibers align the chromosomes along the cell equator. In anaphase, the spindle fibers separate sister chromatids into two separate groups of chromosomes, pulling them toward the poles. And in telophase, the nucleolus and nuclear membrane reform. The chromosomes disperse in the chromatid. Cytokinesis is division of the cytoplasm. The M phase is complete after cytokinesis occurs.
Okay, last but not least, before we move on, we're going to revise how to identify these stages in the microscope. Unfortunately, whatever you've just seen looks very beautiful, but if you were to look into a microscope, you won't quite see these phases that clearly. Can you tell what phase is what? Okay, let me teach you how to find out which phase is which. You help yourself also. Go to Google. Go and type mitosis microscope images. There, they should give you some examples. And the best example to find is this particular one over here. I like this one the most. Okay. Can you try Google that now? Go and Google mitosis microscope images. Find this particular image. I've used this a lot of times as my own reference when I want to identify real life images of mitosis taking place from phase to phase. Okay, can you Google this image? Copy paste into your OneNote. It will help you a lot in being able to identify phases. Once you put that into your OneNote, then you can label some of the defining things that I tend to use to identify the phases. So first one, interphase. When you look at cells, how do you tell they're in interphase? When you look at that circle over there, very often you can usually tell that there is a nucleus still present. Defined by that circle. Other than that, in interphase, the DNA has not yet condensed very much yet. So you find that it exists as a lot of dots. Like, I don't know, what's that, like a hairy ball. Okay, that's what interphase looks like. Prophase is interesting. What happens during prophase? Well, condensation of the chromosomes to become more compact. So what you compare the interphase and prophase. This is the early part of prophase. This is towards the end of prophase. Do you notice that between here and the original state, now you can start to see chromosomes appear? Yeah, it's very obvious now. It's very thick, like worms like that. So for myself, if I can see worms, please don't write this down in your answer. But if you can see like very thick gummy worms, that means prophase has already occurred. You can sort of tell if it's early prophase or late prophase by how dispersed the worm-like structures are. Notice that over here, it is still kind of in a circle shape because the nucleus just broke down. Not yet enough time to disperse yet. But over here, it's starting to disperse already because all of these spindle fibers are starting to pull it apart. Next phase is the metaphase. If I write metaphase, if you know how it works, you'll find that all your chromosomes are aligned like that at the equator of the cell. You look at that metaphase over there. Wow, doesn't quite look exactly like what I'm showing on the whiteboard. Okay? I want you to observe carefully. Do you see that rather than these X structures, you can sort of see a lot of the worm structures are kind of arranged in this format. The V is pointing inwards. Okay, you can sort of see, in this case, hold on, can you tell where the equator is based on how the V is pointing? In this case, the equator is horizontal, not vertical, right? Sorry, how yeah? Okay, oh yeah, that's a great question. Where is the centriole? Isn't the centriole responsible for shooting out all those spindle fibers to be able to align all the chromosomes? How come I cannot see them? Okay, so I'll let you know, when you look under the microscope, you cannot see the spindle fibers or the centrioles. They are too small. The only thing the microscope can catch, right, using just pure light, are the chromosomes. That is large enough for you to see. But you cannot see the spindle fibers or the centrioles. Next, anaphase. When you look at anaphase, you observe that all of these V shapes are then pointing upwards. Okay, so let's go to anaphase. In anaphase, you observe that the V shape is kind of pointing upwards. That's the sister chromatids being pulled apart. Last but not least is telophase. In telophase is when the DNA starts to decondense. Then notice we have two spots here. And my chromosomes don't look quite like the worms anymore. They're starting to look very unwormy-like because they're starting to decondense. I don't need such a compact structure anymore. I'm going to start to reform my nucleus anyway. This is usually how I will try to identify the phases under a microscope. But with that in mind, if you were to look at this image again, can you broadly tell which phase is which? These images, by the way, are not formed using microscope. They are formed using an even more powerful microscope. It's an electron microscope. And that's why, if I were to use an electron microscope, you can actually see some spindle fibers. But you only see that with an electron microscope. But can you tell which phase is which from these four images? Some of them I'm very certain is the phase. Some of them I'm not very clear, even myself. But I can make an educated guess. Which phase are you most confident is the phase? Personally, the most confident one I have is the D one. D, I'm most confident what phase it is. I don't know what about you. Okay. What phase do you think D is in? Yeah, I think it is meta because from what I observed, the chromosomes seem to be somewhat lined up in the middle. Okay, so this is out. That means I'm left with three more phases. Let's look at D, C, and D. Okay, B, C, and A. A and C actually look very similar. They kind of are separate. What's the key difference between A and C? Sorry, Charmaine? Nucleus, okay. When Charmaine observes this, she noticed for C, it looks like there are already two structures surrounding it. Maybe nucleus formed already. So if I compare A and C, I think C is more likely to be telophase because two nucleus has formed. Then if that's the case, what in the world is A? Okay, then the next best guess is A likely came before C. Yeah? Although you don't quite see these V-shaped structures, this is our best guess now. Likely is anaphase, but I suspect it's very, very late anaphase. That's why you don't see those V-shaped structure anymore. Look like they're going to form nucleus very soon already. And what I know is B. To be honest, I'm not very sure. Perhaps the best guess we can have now is prophase. Perhaps it's prophase. I sort of can see worm-like structures. Yeah, I don't see any particular arrangement in the equator. So prophase may be my best guess. Okay? Yes? B and D, is it? Okay, when you look at B and D, notice that, to me now, when I look at D, I can sort of see that there is a line too. Okay? And I think another better sign is that when you look at D, only for this example, because we're using an electron microscope, you can see the spindle fibers arranged in this fashion. Yeah? If you were to look at a very typical structure, the spindle fibers are all aligned like that when all the chromosomes are in the middle. Okay? But if you just use your microscope, actually you will not see all these structures. Okay? Prophase, the chromosomes tend to be more spread out. They are not so aligned in the middle of any equator. So this is an example of a question that may ask you to identify the phases. Okay, so it's a skill to pick up. Some phases are easier to identify than others. Okay, I have around 15 more minutes, so I'd like to spend the last 15 minutes revising meiosis. I mean, I'm assuming you've done SLS, based on what I checked, around 80% of you have. So I'd like to still revise before we go into repro. Yeah, just to let you know, our class has less lessons than other classes by around one period because we have holidays that land during our lesson time. Okay, so with that, let's go to meiosis. Okay, a quick video on meiosis. Then after that, I'm going to talk a little bit about a particular term that I just want to make sure you understand before we go to the next chapter.
In this lesson, we'll explore the details of what happens during the phases of meiosis. Meiosis, sometimes called reduction division, is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into two stages of cell division, called meiosis 1 and meiosis 2. Meiosis 1 has 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. And meiosis 2 also has 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Let's look at what happens during meiosis 1. Prophase 1 starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, one from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatins of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatins in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatins from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happens during crossing over. Then, spindle fibers disappear, and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis I ends with two genetically different haploid daughter cells. Each haploid cell contains only one set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis II. However, unlike meiosis I, DNA does not replicate before meiosis II begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis II ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis I and meiosis II. During meiosis I, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids.
Okay, so let's sum up meiosis. What's the main question? And one of the main reasons why I want to recap this chapter first is because it relates very closely to the next chapter of reproduction. You notice that at the end of meiosis, you get 4 cells. And always from one cell. In one cell, you end up getting 4 cells. And each of the cells contain half the number of chromosomes than when we started off with. Right? The question is, why do we need to have half? What's the point? Well, it relates to the next chapter. Because bear in mind, the process of meiosis is used to produce sperms and eggs. And we need the sperms and eggs to only contain half the number of chromosomes. Now why? Or we think the other way. What if each sperm and each egg has a full set of chromosomes? Will we encounter a problem when mommy and daddy come together? Okay, makes sense now? Because half and half can come together eventually to form something called a zygote. You get a whole. Yeah? You get your complete set of chromosomes again. And so when we say human beings have every cell that you have in your body has 23 pairs. 1 pair came from your mom, one pair came from your dad. Okay? And that is where each one of each pair comes from. That's why you need half also. And that's why meiosis is so important. Only to produce sperm and eggs. So then my next question is, where does meiosis take place in your body? Logically speaking. Yeah. Okay, so Alpha, thank you. Meiosis takes place in the reproductive system. You cannot expect an eyeball cell to suddenly carry out meiosis and then sperm shoot out from there. Okay, doesn't work that way. Sperms and eggs produce at the ovaries, the testicles. So you expect the cells over there to be able to carry out meiosis. Meiosis doesn't take place everywhere in your body. It will be very weird. Sperm shoot out from here, egg come out from your knee. Okay, no. Mitosis takes place across a lot of parts of your body. You will get a cut. Someone punch your eyeball. Okay, then your eyeball cells need to carry out mitosis to replace those that were damaged. Mitosis is for that purpose. For growth, to replace damaged cells. We need cells to be identical and the same. Okay, but meiosis, we use it to produce cells that have half number of chromosomes. Specifically sperm and eggs. Yes? Yeah. A very interesting thing about when we look at meiosis, when we look at your cells, is when we look at your chromosomes, there's this particular word that was introduced inside the chapter. I'd like you to pay attention to. The word is homologous chromosomes. Okay? Earlier on, you notice that when you look at your chromosomes, they come in pairs. Yeah? So here I have one pair, here I have another pair, here I have another pair. But what makes a pair a pair? Number one, if you look at the length of the chromosome, the length of this chromosome is the same as the length of this chromosome. This is a pair. This is a homologous pair. The word homo, same. It means same. So here I have a homologous pair. Here I have another homologous pair. Here I have another homologous pair. I have 3 homologous pairs on the whiteboard. Okay? Other than the length, there's one final thing that makes a homologous pair a homologous pair. Is the kind of genes present. I said earlier on, a pair will contain the same genes. For example, I'm going to represent the alphabet A as the gene for eye color. Okay? Alphabet A, the gene for eye color. Alphabet B, I'm going to use it to represent the gene for maybe your hair color. A homologous pair contains the same genes. Okay? This is for hair color. This is for eye color. G4. G4. Okay, the next thing I'm about to say gets confusing sometimes. So I need you to be all ears. Homologous pair will have the same genes. However, they can have variations of the same gene. Okay, I say again, a homologous pair of chromosomes must have the same genes, but they can have different versions of the same gene. What do I mean? This person could have a gene for brown eye color, so big A maybe is brown, but at the same time, on the other homologous pair, it also has the gene for eye color, but rather than brown, it is small a represents black. Same gene, but slightly different versions of the same gene. Okay, do you catch the ball? I say it's the same gene because you have the same gene for eye color. If I take away the alphabet A, you won't even have any color in your eyes. So the alphabet A represents the gene for eye color, but on each chromosome you can have a slightly different version of this same gene. you can have the black version or the brown version. How yeah?
So the both of the parents have brown eyes, there's a small chance that the child may have like
Okay, so how yeah is skipping a few steps. Okay, now. Therefore, do you notice I can have a few variations of this? Here, I have big A and small a. But another individual for eye color may have instead, rather than big A, small a, they may have instead small a, and small a, small a. That's another combination. Another person may have instead a combination of big A, and big A on both sides. This relates to the two chapters down the road. Because do you realize that therefore, then how do you tell what color eye will this person have? If this person has two of the brown version of the gene, on the surface this person has brown eyes. If this person has two of the black version of the gene, they will have black eyes on the surface. Then the most interesting question is, what if someone has one copy of each? What color eye will they show? One eye red, one eye brown, one eye black? Okay, no, it's not like that. Okay, I'm going to give you an example of a real life example. How many of you here have blood type AB? Okay, Mercy have, Finia have. You know, people with blood type AB, you have a pair of chromosomes that determine blood type. People with blood type AB, they have A and B in their cells. Who has blood type O? People with blood type O, rather than A and B, you will have just O on both sides. Okay? So I'm just teaching a few steps ahead because I want you to appreciate the idea of homologous chromosomes. That even though we say it has the same genes, it can have different versions of the same genes. And rather than say so much, the different versions are just alleles. For every gene, there are different alleles. Okay, and I need to cut 11:30 already. Yes.
how about the people with different eye color?
Different heart.
Different eye color.
Oh, we will come to that chapter, then we'll talk more. Okay, thank you. Okay, so the next time I see you next week, we are going to start new chapter reproduction. We're going to learn about your body parts. Okay, don't need to be shy, although I myself